All right, in the last section, what we were talking about is populations, and right here now, what we're going to talk about is really how that relates to the Hardy-Weinberg equilibrium theorem that we were talking about a couple weeks ago in class when we were doing the lab. So if we look at the idea of allele frequency, if you consider a population of organisms that's, say, numbers 500, then we would have 1,000 alleles, because each organism has, two homologous chromosomes and then therefore two alleles at each locus. So for instance, if we say that we've got these two alleles, big R and C big W. R stands for the red and W stands for the white. You can see that if we have CR, then we've got red, CW, we've got white, and CW, then we would have pink, okay? And on our breakdown, and for this hypothetical breakdown that we have, we're just applying some numbers here. If we have 320 of them that are red, CR, and we have 160 of them that are pink, CW, and then 20 of them that are white, what we would suggest from this is the blending hypothesis. But as we learned in the last section that we were talking about, with this blending hypothesis, what we would actually be doing is eliminating variation. And we don't have blending. We can't use blending because it eliminates the variation. And if we set up the actual Punnett square, for instance, like if we just say, whoops, let me get my pen here. If we do CR, CRCW, and then CW. If we take the two pink, we get, let me undo this here real quick. In our Punnett square, we would have CR, CW, CW, and CW. So if we took two pink, this one of course is pink, and we crossed it to this one, which is pink, we would get red and white as well as some pink. So we would get these parental offsprings again. So that's why we can't have blending. It's going to eliminate variation. And then in addition to that, if we cross two pinks, you would expect maybe to get pink if you thought about blending. And when you actually do that, you get the red and the white back. So that's why it can't be blending, okay? So when we learned about the Hardy-Weinberg theorem a while back, what we were basically saying is this is a way to, for us to look at allele frequencies and see if they're going to change over time. And I guess the key thing here to keep in mind is that this is when independent assortment and segregation alone are working. So in other words, none of those other 5 things that we'd talked about in class were working on the alleles. Just independent assortment and segregation. And those 2 things alone really kind of apply to a non-evolving population of organisms, okay? So in other words, there's no natural selection, no evolution occurring. So then what we're saying is the allele frequencies would remain the same, okay? And we talked about the reasons why, of course, this doesn't happen, and we'll get into that and revisit that here in a minute. But what the Hardy-Weinberg theorem is doing is explaining really a non-evolving gene population, okay? These frequencies of the alleles should not be changing. And if they are changing, then of course it says that evolution is occurring for one reason or another, okay? So in other words, sexual reproduction alone will not change the frequency of the alleles in a non-evolving population, because if you recall from the meiosis stuff that we learned about last year, segregation, independent assortment, those types of things are what's really shuffling the genes aside from all of the crossing over and the other things themselves, okay? So what this theorem basically is doing is looking at Mendelian inheritance, and what we're looking at again is the non-evolving population, and it helps us understand this long-term evolutionary types of the change that's occurring, okay? So this genetic variation is basically what's going to allow natural selection to actually occur. So if we look at our population again, we've got the 1,000 copies of these alleles, and the way this Hardy-Weinberg theorem essentially works here is, let's say that we have 800 of them that are C big R and 200 of them that are CW. So if we go back here for just a second, if you look, we have 320 red, 160 pink, and 20 that are white, okay? So 320, CR, 160, CR, CW, okay? And let me do this real quick. And then 20 of them that are CW, CW. So just for you guys to be able to see where we get these numbers from, there's 320, 320, that's 640, plus 160 gives us 800, okay, CR alleles, and then 160 plus 20 plus 20 is 40, which is where the 200 CW comes from. So that's where these numbers here are coming from, okay? So now if you're looking at this, we have 1,000 total alleles, and this represents CR, as we said, which is equal to P, or the dominant allele. CW is equal to Q, which is the recessive allele. So 800 out of 1,000 equals 0.8, and 200 out of 1,000 equals 0.2. And that's where these percentages kind of come in, okay? So if we're looking at that, if we want to look at the probabilities now based on what we'd learned before, when you have this P squared, okay, what you're basically saying is CR, CR. So we know the frequency of P equals 0.8. So 0.8 times 0.8 equals 0.64, okay? And then Q squared equals CW, CW, and we know that that is 0.2 times 0.2, and this is 0.04, and then you could add these up and then subtract, or you can just do 2PQ is equal to the CR, CW times the 2 there. So this you have R 0.8 times 0.2 times 2, which is 0.32. Okay, so when you add all of these up here, this is going to give you the 1, or 100% of the actual population. So that's what we're looking at there when we're determining the allele frequencies and percentages. And we'll get some more practice with this in class. Okay, so here's the Hardy-Weinberg equilibrium equation, of course. You have P squared plus 2PQ plus Q squared equals 1. And in a non-evolving gene pool, what you're going to have is, if you have this random mating, no matter what you do, the frequency of these alleles is always going to be remaining the same, okay? And if any of those 5 things here that we've discussed in class, the large population, no gene flow, new mutations, random mating, and no natural selection, if this is what's actually happening, then that's where you have Hardy-Weinberg equilibrium. And if you get a departure from any of these 5 conditions, that's where you can say that evolution is actually going to occur. So in other words, you might have a large population, but the gene might be flowing well through there. Mutations, of course, are happening at random. You've got non-random mating, in other words, the choosiness of one sex for another, for, like when we talked about the trains on the peacocks, we talked about the frigate birds with the large red air sacs below their beaks. We talked about the lion's mane. All of those types of things leads to choosiness within mating systems. And then, of course, the organisms are interacting with their environment, so therefore we have natural selection occurring. So these types of things is really why Hardy-Weinberg doesn't hold true, and why we can say, of course, that populations do change through time, which, of course, is evolution. Okay? So as far as mutation, sexual recombination, that sort of thing goes, these types of things provide for a lot of variety within the gene pool. We discussed this last year with meiosis. We talked about it a little bit in class this year. And what these mutations basically do is they change that nucleotide sequence. They give rise to new genes. These new genes, whether they're good or bad, really depends on the interaction with the environment and how the environment's changing. Oftentimes they're not good. Sometimes they are, it just, again, it largely depends on the organisms and the environment that they're living in. And you have to keep in mind too that many mutations that we're kind of familiar with arise in somatic cells and never get passed on that way. Only the types of mutations that arise in gametes are going to have an impact really from an evolutionary standpoint. And really the odds of a mutation arising in the I guess the gamete, the sperm, the egg, and then actually getting passed on is relatively low. So that's why, the mutation rate, generally speaking, for larger organisms is quite low. If you think about a smaller organism, they generally have a very rapid generation time, and they have lots of mutations. They don't have the proofreading mechanism that the larger eukaryotic types of organisms have, and so forth. So plants and animals generally have long generation times, and they have a much lower frequency of mutations than something like a bacterium or a virus or a yeast or something like that, where the mutation rate is usually a little bit higher because the generation rate is so quick. Okay, so sexual recombination, crossing over in prophase 1 of meiosis, this is where we get a lot of variation occurring on a generation-to-generation scale. And the three main things that we need to kind of understand here from an evolutionary standpoint as far as altering the allele frequencies would be natural selection, gene flow, and genetic drift. So with natural selection, there's a lot of things that we have to understand that we talked about in class and how natural selection plays a role in evolution. We talked about the organisms, the traits, the adaptations, the variation that they have. Some of this variation makes them better suited for their environment, and therefore they're more fit to survive. And we described fitness as being that thing which really is a measure of the reproductive success of the organism. And then the adaptation characteristics that they have that make this individual better suited to its environment, its way of life, attracting a mate, that sort of a thing, is what's going to lead to really the success of the organism. The survival often depends on the change in the allele frequencies within the population. And we have to keep in mind that natural selection is what's acting upon that variation. And within members of the same species, those organisms that have those traits that allow them to better seek out a mate, avoid predation, do all of those types of things are the ones that are going to be more fit and therefore going to be really more favorable in terms of evolution, and that's what's going to allow that population to change to kind of reflect those traits. Okay, we talked about the notion that species often produce more offspring than the environment can support. We talked about producing lots and lots of minnows and fish because very few of them survive. We talked about sea turtles. We talked about really plants producing many offspring because only a few of them are actually going to end up really, say, as trees or as other plants and so forth. So the individuals that are more prolific and more hardy is in terms of like their ability to withstand the environment, these are the ones that are going to produce more offspring over time, and those members of the population are going to be the things that are going to survive, whereas the other ones are not. They're going to die out along with those genes that aren't suited for that particular environment. Okay. So, of course, individuals, they're passing this reproductive success on, and what might be beneficial now may not be beneficial in the future. So we have to kind of keep that thing in mind too, as the environment changes, what might be deemed good for us now, good for the population as a whole now, may not be, say, in the future. Okay? And we talked about the rock pocket mice being brown, blending into the environment was a good thing, but 1,000 years ago in New Mexico when the volcanoes erupted and put the black lava down, those organisms that had the black mutations, which were once disadvantageous, in other words, they stuck out on that brown background, those things were now able to camouflage themselves, or they were camouflaged rather, on this black rock, and then they became more successful, and their population exploded, and the brown ones remained in the brown environment, the black ones remained in the black environment, and therefore, because the environment changed, certain traits therefore became favored. Okay? So natural selection really is going to increase the frequency of the characteristics that make these individuals better adapted to their environment, and we said, and of course decrease the ones that don't. So now genetic drift, this is another thing here. This is an unexpected type of a fluctuation in the allele frequency from one generation to next, and it's basically due to some chance event that wipes out a particular population, whatever it may be. A windstorm, a thunderstorm, a hailstorm, just something that happens to, for whatever reason, wipe out a large percentage of the population or all of one particular type here, and the gene pool really drifts to kind of reflect this. Okay, and there's two situations really that increase the likelihood of genetic drift. You have the bottleneck effect and the founder effect. And what happens when you have a sudden change in the environment is the survivors sometimes pass through what's known as a bottleneck, and they don't represent the individual population as a whole. So if you have your kind of ancestral population right here, you can see that there's a pretty equal mix of blue and white, it looks like, and then you've got some yellow sprinkled in. But when you have this event taking place here, here's this so-called bottleneck where these things are passing through. And if we look at the population after it passes through the bottleneck, you can see there's a large percentage of blue compared to the white there, and there's no yellow at all. So we say that this population has gone through a bottleneck, and now the survivors here in 2 don't reflect that original population. Okay. When the few organisms become isolated, now they establish this new population. This is where we have what's known as the founder effect. So they can pass through a bottleneck. So if this original population here in 1 didn't die out, rather this population right here was separated from it, we can say that they are founding a new population. So we say the founder effect has occurred. They've passed through this bottleneck, and now their gene pool is not representative of that original gene pool. Okay, so this could be, for instance, when we talked about the beak of the finch, one of the hypotheses is that an original ancestral flock of birds from the mainland of South America, a small portion of them got blown off course somehow, and therefore they started to kind of adapt and radiate out into the archipelago that is the Galapagos, and they changed dramatically from that original population. So that population that originally made it to the Galapagos Island were the founders of this new population, so to speak. Okay. Now, gene flow, this is when populations gain or lose alleles as a result of the migration of the organisms in and out of the population. And what this tends to do is really reduce differences between the populations. In other words, they're bringing genes in from various populations surrounding, so then they all begin to kind of interbreed with one another and mix those genes, and that's what we refer to as gene flow. Okay, so variation, of course, these are the heritable differences within the populations. These are the things that comprise the raw material, really, that natural selection is going to act upon, because that variation is sometimes good for the environment, sometimes bad for the environment, and that which is good gets weeded out from that which is bad, and so forth. And it's only the genetic component of this variation that has any evolutionary consequence at all. Okay, so it has to be some sort of a heritable characteristic, an allele, something on the genes that can get passed from one generation to the other to have any meaningful impact on the evolution of the organisms. Okay. And what you have here with this variation is you have these discrete or these quantitative types of characters. And sometimes we refer to them as continuous or discontinuous. So this discrete characteristic would be an either-or basis. You're either tall or you're short. You're black or you're white. There's no in-between in terms of the type of organism that we're talking about. And this would be discontinuous as well. Okay, and I guess the tall and short thing really wouldn't be the greatest type of an example, because really with you could look at it as either tall or short, but we know actually that the height rather is a quantitative or a continuous type of variation, which really kind of spans a range from being short to being tall. So in other words, you're not one or the other. You can be any of the in between. So we talk about discrete or discontinuous, okay? And we talk about quantitative or continuous. And usually with quantitative, as it says here, is you've got 2 or more loci that are responsible for determining the type of phenotype. So skin color and in height are 2 examples of quantitative or continuous types. And then discrete, this is this either or basis. And you see it with like flower color. You're either white or you're purple, for instance. There is no in between, okay? Now, as far as the variation goes, these variations can arise, of course, we talked about in class, from mutations that occur during meiosis. Meiosis itself can give rise to this variation with this crossing over. The whole process of sexual reproduction can give rise to a lot of variation within these individual species. And this variation is really, again, what this natural selection is going to act upon. Okay, and we talked about the advantage of the organism and this adaptive advantage, the variation that they have that leads to this notion of fitness, which is the improved, I guess, reproductive success, giving rise to more offspring, having a greater contribution on a genetic level to the gene pool from one generation to the next. In other words, that fitness, we said, is the reproductive success of the organism. It's not just how robust it looks, how good it looks, or how strong it is. in addition to that, oftentimes it has to include the idea of reproduction and giving rise to organisms in future generations. Okay, so in the next part 3 here, we're going to talk about the different modes of selection and go through a few videos and things like that. So it's important that you write down questions if you have them, and we'll definitely talk about those in class. Okay, so we'll see you soon.